The cell cycle begins with interphase, which is the process of preparation for cell division. It consists of 3 stages. G1 phase is the first growth phase and is when the cell increases in size. It also begins to synthesize new organelles and proteins. S phase stands for the synthesis of new DNA and is also when the chromosomes replicate. 2 specific genes control cell division during S phase. Proto-oncogenes act like accelerators that stimulate cell growth and division. They work together with tumor suppressor genes that act like brakes to slow down or stop cell growth and division. This creates a good balance to the cell cycle. At the end of the S phase, the amount of DNA has doubled. The G2 phase is the last and shortest phase of interphase and it ensures that everything is ready to begin the process of cell division known as mitosis. The cell spends 90% of the cell cycle time during interphase. After interphase has been complete, the cell enters the process known as mitosis, which is divided into 4 main phases known as prophase, metaphase, anaphase, and telophase. During prophase, the genetic material found in chromatin condenses into visible chromosomes. Centrioles begin to separate and migrate towards 2 separate ends of the cell. Microtubules form between the 2 pairs of centrioles and become assembled into tiny fibers known as spindle fibers. The nuclear envelope begins to break and later disappear. At metaphase, the chromosomes move to the middle and become aligned. Each chromosome is connected by a spindle fiber at its centromere. During anaphase, chromosomes are split and the sister chromatids move apart to separate into the opposite sides of the cell. This results in a group of chromosomes that have been split completely. The final stage of mitosis is known as telophase. This is when the chromosomes gather at opposite ends of the cell and lose their distinct shape. A new nuclear envelope will reform around each set of chromosomes. Cytokinesis begins when the cytoplasm pinches in half, forming 2 new separate daughter cells. Each daughter cell has an identical set of duplicate chromosomes. This is the last phase of the cell cycle before it enters interphase again. However, sometimes radiation, unhealthy lifestyle, smoking, pollution, some chemicals, and heredity are among some factors that stimulate mutations. Mutation of proto-oncogenes result in oncogenes and the mutation of tumor suppressor genes can cause them to become inactive. These mutations stimulate excessive division, causing the cell to divide more frequently than it normally would, and it is known as uncontrolled division. This disturbs the balance and can cause cancer.